Next we have is specification number 3, which says, explain the effect of changes in temperature and pH on enzyme activity in terms of kinetic energy, shape and fit, denaturation and the frequency of effective collisions. So now, we're going to look at how temperature and pH affect enzyme activity, just like we explored earlier. We'll explain these effects by considering things like kinetic energy, how well the enzyme and substrate fit together, the risk of denaturation, and how often successful collisions happen. So let's begin by exploring the effect of temperature on enzyme activity in terms of reaction rates and enzyme stability. Every enzyme has a small range of conditions where it works well. We call this its optimal temperature. That's the temperature where the enzyme is most active and the reaction happens the fastest. Now, if the temperature drops below this level, everything slows down. The molecules don't move as quickly, so there are fewer collisions between enzymes and substrates. As a result, the enzyme can't function properly. But if the temperature rises too much, we run into a bigger problem. The heat can actually start breaking the bonds that hold the enzyme's shape together. When that happens, the enzyme changes shape and can no longer fit with the substrate. We call this denaturation. And once an enzyme is denatured, it can't do its job anymore. If we look at a graph of enzyme activity against temperature, we'd see a curve. It starts off low, increases as the temperature rises, hits a peak around 37 to 38 degrees Celsius, that's the enzyme's optimal temperature, and then drops sharply as the enzyme begins to denature. You'll usually see an arrow pointing to that peak to show the optimal temperature. Its shape is what allows the enzyme to do its job properly. Each enzyme has a very specific 3D shape, and that shape comes from the sequence of amino acids that make up the protein. One part of this shape, the active site, is especially critical. This is the region where the enzyme binds to its substrate, and it has to be a perfect match, like a key fitting into a lock. Enzymes also have an optimum temperature. For humans, that's around 37 degrees Celsius. That's the temperature where they work best. But when the temperature goes higher than this, things start to change. As we heat an enzyme beyond its optimum, the bonds that hold its shape begin to break. That means the enzyme, especially its active site, starts to lose its exact shape. When this happens, the substrate no longer fits properly, and the enzyme becomes less effective. If the temperature keeps rising, the enzyme becomes denatured. That means the active site is permanently damaged and the substrate can't bind at all. Once an enzyme is denatured, it can't catalyze reactions anymore, which can seriously slow down or even stop important processes in the cell. A simple example of this is when you cook an egg. The clear egg white, made of proteins like albumin, turns solid as it denatures. This kind of damage to enzymes can have serious consequences for living organisms. If enzymes stop working, cells can't function properly, and most organisms can't survive temperatures above 50 degrees Celsius for very long. We can also test for denaturation. If we boil a substance that contains enzymes and then try to use it in a reaction, and nothing happens, it likely means the enzyme has been denatured. But not all enzymes are that sensitive. Some heat-resistant enzymes, found in certain bacteria, can still function at high temperatures, even above 50 degrees Celsius. These enzymes are really useful in industry. For example, they're used in biological washing powders, where they help break down stains in hot water. And research is ongoing. Scientists are constantly exploring new heat-resistant enzymes that could be useful in all kinds of industrial applications. Now, if we look at the diagram, we can see how this process unfolds visually. On the left, we see the enzyme and its substrate fitting together perfectly. That's under optimal conditions. In the middle, the temperature is starting to rise above the optimum. The enzyme is just beginning to distort, but it might still function. Then finally, on the right, we see a denatured enzyme. Its active site is completely misshapen, and the substrate no longer fits. That's what denaturation looks like, and why shape really does matter when it comes to enzymes.